Cells reproduce asexually through a series of processes. The first one is called interphase, then mitosis, and finally cytokinesis. The cell division is regulated by cyclins. However, sometimes the DNA in a cell is defective, and it starts to divide exponentially and starts to form a tumor. This is the start of cancer. Here are all the parts of the cell. The nucleolus controls the cell. The nuclear envelope holds together the nucleus. The cell membrane holds together the cell. The chromatid are the messy genetic material, and the centrioles help organize the chromosomes. These are chromatids, which are condensed chromatin. These are chromosomes, the visible X-shaped genes consisting of two chromatids joined together by centromeres. The first process is interphase. It takes up 90% of the duration of the cell division process. G1 or GAP1 is the first stage of interphase. It is when the cell grows to make room for the new proteins and organelles that are going to be formed during the S and G2 phases. The S phase or synthesis phase is the second stage of interphase. It is when new DNA is created. The chromatin within the nuclear envelope doubles so that when the cell splits, each of the daughter cells have the same amount of genetic information in them. In the case of cancer, most cancer cells have a defect in the gene called p53, so the cells cannot gauge how much they have to grow and when they have to divide, so they divide exponentially. The genes within the cells are proto-oncogenes, genes which encourage cell division. However, when they mutate, they become oncogenes, which divide excessively. These cells can cause cancer. The other genes are tumor suppressor genes. They discourage cell division, and they can become inactive when mutated. G2 or GAP2 is the final stage of interphase. It is when the cell fully prepares itself for division. It is the shortest phase. In interphase, all of the necessary molecules and organelles are created during the stage. After this, the cell is finally ready for division. The second process is mitosis. Prophase is the first stage of mitosis. It makes up half the time for which mitosis occurs. It is when the chromatin within the nuclear membrane condense and become visible as the X-shaped chromosomes. While this is happening, outside the nucleus, the mitotic spindle starts to form. The function of the spindle is to pull apart the centromeres connecting the chromatids. Also during this stage, the nuclear envelope disappears. Metaphase is the second stage of mitosis, and it is the shortest. It is when the centromeres connecting the chromatids form a line through the center of the cell. The mitotic spindle fibers joins up the centromeres to the poles on opposite sides of the cell. Anaphase is the third stage of mitosis. It is when the mitotic spindle fibers separate into two chromatids that were forming the chromosomes, and then they move along the fibers to the poles of the cell. Once separated, each chromatid is considered to be a chromosome. Anaphase comes to an end once the chromatids have moved, and the mitotic spindle fibers disappear at the end of the stage. Telophase is the last stage of mitosis. It is when the tidy chromatid unwind into chromatin, the messy string-like structures. The nuclear envelope